Imagine that you are a patient who has just been recently diagnosed with cancer. As devastating as this news is, there are several available treatment options for cancer. One such treatment option available is chemotherapy. Chemotherapy drugs actively target cells during different phases of the cell cycle in order to stop the development of new cells from growing. This treatment has been found to work because cancer cells have been found to grow and differentiate faster than regular cells do. One major problem with cancer cells is their ability to travel and migrate throughout the body, such as through the lymphatic system or the bloodstream. This movement can lead to tumor metastasis, which is cancer cells moving from the original tumor site to other parts of the body. But what if there was a way to target these cancerous cells? Would this help in the localization of cancer, and subsequently, would this help in the removal of the isolated tumor? Actin cytoskeleton has long been known to be the major source of internal force created by the cell. It has been shown to be involved in cell movement as well as cell shape. The movement required to travel around the body involves the interaction of several different cytoskeleton proteins. Examining the different levels of cytoskeleton proteins involved in actin turnover during migration can give greater insight into the protein interactions that are involved with cell movement. My current research interest is in understanding the underlying mechanisms of cell movement. My model of study is a monocytic leukemia cell line known as THP1. These cells are undifferentiated monophages, and injecting them with a chemical that induces differentiation, like PMA, allows them to differentiate into mature macrophages that can then travel and move about in their environment. Assessing the levels of various proteins involved in actin turnover following differentiation can allow for a greater understanding of the cell movement. Proteins involved in my research include WDR1, cofilin, and CAPS, which is cyclase-associated protein. These are all proteins that are involved with actin turnover. The current research underway has both basic and applied routes for potential cancer treatments. By understanding the mechanisms of cell movement through cytoskeleton proteins involved in actin turnover, there is the potential ability to restrict the movement of cancer cells throughout the body.